Imagine walking into the doctors and being told you have cancer. It's not something any of us want to hear. But is it a relatively harmless lump, easily removed and forgotten about? Or is it a potential killer version requiring more aggressive treatment? Traditionally, cancer diagnosis is based on anatomical location and a pathology assessment of the stage or aggressiveness of the tumour. This guides whether a patient receives radio or chemotherapy or not. But the treatments are standardised for a particular type of cancer. Now, diagnosis and treatments have definitely improved over time, but they don't always work. And why do patients respond so differently to the same drug for the same type of cancer? Why doesn't one size fit all? Now, 25 years ago, I was a young scientist working at the National Institutes of Health in the US when James Watson, he who won the Nobel Prize for defining the structure of DNA, announced to a stunned audience the launch of the Human Genome Project. Now, this was a quest to sequence and decipher all 6 billion chemical letters in the human genome. This is the DNA that is so nicely contained within all of our cells, as Antoine had very clearly explained earlier, which holds all the instructions needed for life. Now, at the time, this Human Genome Project was audacious as sending a man to the moon. The methods for sequencing were manual and highly laborious. For example, it took a week to sequence just 1,000 letters long. So imagine for 6 billion letters, it was projected to take 15 years, $3 billion, and thousands of scientists slaving around the clock to achieve this aim. These days, it takes just 1 week and $5,000 to sequence an entire human genome. But taking all of this complex information and turning it into something meaningful or clinically useful is the next big challenge. Having said that, there are some genetic tests of inherited diseases now available. Think of the breast cancer gene made famous by Angelina Jolie. But it's more complex than that. Most cancers aren't inherited and aren't due to one single mutation in a gene. So we know now from sequencing of many cancer genomes that even cancer cells that look the same under a microscope are actually quite distinct at the genetic level. And this may explain why one size does not fit all in cancer medicine. So here we have a group of patients that have the same type of cancer. But the genomes of these cancers harbour many different mutations or DNA alterations. We refer to these as cancer genetic markers. These markers are like personal signatures or identifiers that appear to be able to distinguish in which patient these cancers are more likely to spread or to predict risk. The genomic information that is gained from these signatures or these markers can also be used to guide the treatment that is best suited to each patient. So, okay, what if we can use these different genetic markers to come up with good versus poor gene signatures to help predict risk? So cancers that are harmless or benign lumps would have a good signature. And this is associated with good prognosis and responsiveness to standard therapy. Poor signatures, on the other hand, would signify cancers that are at high risk of invasion and spreading to other sites in the body. This is a process known as metastasis. This is linked to poor prognosis because it is metastasis that typically kills. And furthermore, there may be within these poor signatures particular mutations that we know make those cancers unresponsive to chemotherapy. So it would be much more effective if we could target those particular mutations with specific drugs. Now, this would spare those patients unnecessary chemotherapy that we know doesn't work for them. So this is the premise behind personalised medicine. And there are working examples of this. There is a simple genetic test that identifies breast cancer patients for responsiveness to a new drug called Herceptin. So what are we doing that's new? Well, we are studying a very, very common skin cancer, but not at all well understood at the genetic level, called cutaneous squamous cell carcinoma. Now, given that 2/3 of Australians will have had a non-melanoma skin cancer by the age of 70, chances are that many of us in this room would have had one frozen off by their GP. Now, I know I have, and I'm not even 70. Believe it or not. Now, most of us have thought nothing of that, nothing more of that, and that is fine, because of the at least 300,000 or more cases each year in Australia, most are superficial, harmless spots. But a small proportion of these, especially of cutaneous squamous cell carcinoma, will spread. It is these metastatic skin cancers that we are focusing on because we just don't know what makes some skin cancers metastasise and others not. So in this example slide here, this is a specific case handled by a surgical colleague of mine. This rather innocuous-looking spot, I don't know if you can see it or not here, didn't look so great. It was removed and examined and looked for all intents and purposes like a low-risk or non-spreading version of skin cancer. So no further treatment was deemed necessary for this patient. Unfortunately, this patient later presented with metastasis in the lymph nodes in the neck and required extensive head and neck surgery to remove the involved and surrounding tissue. And now this can be quite disfiguring and have further morbid consequences. And apart from post-operative radiotherapy, there are no standard or approved chemotherapy treatments for metastatic skin cancer. So if this cancer has progressed beyond that local site, there are no approved treatments for that type of cancer. So prognosis would not be great. By the way, my colleague alone handles at least 80 such cases a year in the Illawarra, and mostly men, by the way. So what if we could more reliably diagnose patients, such as this one, at this early stage back here, when it wasn't even obvious that this was going to metastasise, and then target them for closer observation and/or more appropriate treatment? An earlier diagnosis would have helped this patient immensely. So our big idea is to do just that, and we are adopting whole genome sequencing methodologies to achieve this. So we start by collecting tumour samples from metastatic lymph nodes and from blood from local skin cancer patients. We extract and process the DNA and send that away for sequencing. Then the genomes from the normal, that is from the blood and from the tumour samples, are compared to each other. And we use the normal samples to look for mutations that are unique to the tumour and not just a normal variation or harmless variation found in the normal samples. So if you imagine that these letters here are a DNA and the phrase makes up a gene, we're looking for the differences in the tumour DNA or the cancer DNA that are not present in the normal sample. And by the way, there is a duplication that's present in the normal, it's also present in the tumour. That is probably just a harmless variation that's specific to that patient. So once we have a library of cancer genomes assembled, well, we need then to try to decipher all of this genetic information by comparing recurring patterns, or these gene signatures of each patient, with how they are faring over time. So have these patients progressed or not? And hopefully we can see by whether they have a poor or good signature that we can predict the risk of them having metastasised. So to this point here, this will represent a major achievement because we will have gained an understanding of the genomics of metastatic skin cancer, because at this point we don't understand it. So that would be really great. But the bigger picture for us would be to use this information to develop personalised treatment plans. So imagine if a gene signature can be converted into a fast and cheap test that your GP can use as a reliable guide to decide whether or not just to get out the liquid nitrogen gun and then send you on your way, or to refer you for more proactive and hopefully more targeted treatment as soon as possible. This should reduce the often devastating surgery and poor prognosis currently associated with metastatic skin cancer treatments. So we really do hope one day to be able to personalise metastatic skin cancer medicine for a more tailored approach. Thank you.